This conference will now be recorded.
All right, very good, folks. So this is chapter 3, chemical level of organization. We're going to start on this chapter here. Again, this is going to be very different from the other PowerPoints and lectures that I do, in that this is just kind of like a review of important information that you'll have to know for the test coming up for test number 1. Now let me find for you, we're going to start. All right, so here's elements and atoms, the building blocks of matter. So here we go with slide number 1. So I'm going to be using both slideshow here with you all and recording this here. So matter, mass, right? So understand that as far as when we're talking about chemistry and we're talking about the fundamentals of chemistry and such, right? So look at this here. Anything that occupies space has mass, right? That's matter. Mass, the amount of matter contained in an object. Now, mass of an object is not the same as the weight of an object, because weight, what's the factor with weight that can make variables? Gravity. Understood? So the mass of this object right here, so we have here like this, little stapler and such, right? Depending upon where it's located, the weight can change as a result of gravity, gravitational forces placed upon it, okay? So you got to keep that in mind as far as gravity and mass and matter are not the same, okay? So weight can vary because it depends on gravity. Now I'm going to there we go. We're going to the next slide here. And again, I've taken the time to highlight this for you all. So I've looked at the chemistry notes study guide that Professor Massimo had provided for you all. And so I'm taking this with our PowerPoint and just highlighting in particular, you all, so understand this, folks, you are all responsible for the chapter. You're responsible for the chapter. My job as a professor is to try and condense the information and make sure that you understand what's being in the chapter and such, but you are responsible for reading the full chapter. And folks, you know that really any of the work and the material that we're doing here in class, you should be reading the chapters at least once. Then you have the PowerPoints. Then you have notes that you're taking in class, right? So there's a lot of different ways that you can be learning this information that I'm disseminating it to you. I'm teaching it to you all, right? And trying to make it easy to understand. So matter is composed of elements, right? So you'll see here that pure substances that are made up of a single type of atom. And so you'll see on the right-hand side of this, right, with this table, it's just such an example of certain ones as far as oxygen, carbon, hydrogen, nitrogen, calcium. And understand this, just to throw this out at you, and we'll be looking at this more in the respiratory system in A&P2. Primarily, your atmosphere that you live in and you breathe, the air that you breathe, is primarily oxygen? No, it's not. It's primarily nitrogen, right? So the air that you breathe and the atmosphere that we're living in, primarily it's nitrogen, okay? Oxygen is only like 20 some odd percentage, right? It's nitrogen is we're in the 70 percentile, right? So quite interesting. So anyhow, you've got you've all in grade school have seen the periodic table, right? Periodic table of what? Of elements. Yeah, so elements, pure substances that are made of a single type of atom. What's an atom? Smallest unit of an element that retains properties of that element, right? Simple enough. Molecules and compounds. You'll see here as far as that bonds are taking place and we're combining atoms, we're combining elements in order to form these. Now here we're looking at as far as the subatomic particles, okay? You all probably remember this a little bit as far as protons, neutrons, electrons. Understand this, that the mass of an atom is primarily protons and neutrons. The mass of an atom is primarily protons and neutrons. Electrons, really, really small. I mean, as much as the protons and neutrons are small, electrons are even quite small. And so they don't really they don't really comprise enough mass in order to contribute to it. So understand this, that Protons, again, positively charged, the P and the P. Neutrons, neutral, there's no charge. Electrons are negatively charged. Electrons really are kind of hovering around. So look at this for a moment. So the nucleus of an atom, protons and neutrons, right? All that's rotating around and orbiting around the protons and neutrons would be the electrons, very tiny, but moving around. Here's just an example of that periodic table of elements. And you can see here as far as what are, so this just helps you to having an understanding of when you're looking at the periodic table of elements, what do all the numbers represent and such. And so you'll see here atomic number, and this is the number of protons in the nucleus, right? The atomic number. You're gonna have to know this, atomic number, the atomic weight. Atomic number, the number of protons in the nucleus of an atom. The atomic weight, the weight of an atom is the combination of protons and neutrons, okay? So the weight, protons and neutrons. The atomic number, the number of protons. All right, so here we go. Atomic number, it's just what we just said, atomic number, number of protons. Atomic weight, protons plus neutrons. And again, like I said, you see it says there, small amount of weight from the electrons. It's so negligible that they don't even consider that really being a part of it. So the atomic weight, protons and neutrons. The mass number, right? Roughly equal to the protons and neutrons, number of protons and neutrons. The isotope, isotopes, these are different forms of an element, different forms of an element, okay? So you'll see here that they differ in the number of neutrons, and that's the key here. The different multiple isotopes of an element will only differ in their number of neutrons. So here we go. So here's isotopes of hydrogen. See, so you have here protium, deuterium, tritium, right? So what do they differ in? Their number of neutrons that are present. So you'll see here, protium, deuterium, tritium, the number of neutrons is different in each one of those, okay? Ions. And know this, folks, that in anatomy and physiology, in the study of the physiology of the human body, ion concentration is really a big deal. And you're going to see this as time goes on, as we look at the muscular system, as we look at the nervous system and such. Ion concentration is a big deal. So an ion is an atom with an electrical charge. And so we can have cations and anions. So when you think of a cation, I see the plus sign right there. See the T? That's the plus sign. So what is a cation? They are positively charged. An anion, see the N? Negatively charged. That's how I remember it, okay? So however it helps you, very good. But there's a charge. And so you'll see here, positive or negatively charged. And ions are formed when atoms donate or accept electrons. So that's really the factor there, whether we have an anion or a cation. Now, I'm not going to go over this with you all, to be honest. Really, like I said, in your chemistry class, they're going to review this with you. But understand that really there are shells, there are layers of moving and circulating electrons. Okay, and so we have these shells, right? And the outermost one is called the valence shell. Okay, and really the reactivity of an atom depends upon whether that valence shell is full or whether it's not full, and then it's going to be more reactive. That's the gist of it. I know that sounds like, Doc, I'm not understanding what you're saying, but you read through it, look at it and such, and you'll see that that reactivity of an atom really depends upon what's going on in that valence shell. So you'll see here, atoms react in a manner to completely fill their valence shell. Understood? So if we're missing electrons, then the atom will do all it can. It's going to want to fill that shell, that outer shell, that valence shell, so they can either share, accept, or donate electrons to do so. And here with this next slide here, you're just seeing that So the first electron shell can take 2 electrons. The second one up to 8. And atoms with more than 10 electrons require additional electron shells. You'll see they're represented with number C, letter C there, and sodium and such, okay? Now, bond formation is a big deal. Okay, bond formation is a big deal, and we're going to talk about the different types of bonds that are present, and you'll have to know what distinguishes them and such, okay? But you'll see here that, so with this slide here, it's showing you that atoms react with other atoms to try and fill their valence shell. I said it to you before, right? That they're reactive if the valence shell, that outer shell, is not full. So they're going to want to try and fill it to become stable. Understood? Stable, stability. So you'll see here potassium. So potassium is right here. So potassium has 19 protons. Oh, sorry, come back. 19 protons, 19 electrons, no charge. The electron in its outermost shell is unstable. So potassium, let me move this for a moment here. All right, potassium will give up its outer electron, and now it has 19 protons and 18 electrons. So it has a positive charge, and so it donated. What did it do? It donated an electron. So now there's more of a stability there. And as it is written at, potassium ion would be what? K, right? Because it's kalium, that's Latin and such, would be a positive sign, okay? And you'll see here as far as fluorine. Fluorine has 9 protons and 9 electrons, no charge. Its outermost shell is unstable because it's almost filled, but not full. So fluorine gains an electron in the outermost shell. So now it has 9 protons and 10 electrons. There's a stability there, so it has a net negative charge. It actually received an electron. So you see that the potassium was what? Donated an electron, positive charge. The fluorine gained an electron, right? Gains an electron, and it has then a negative charge. And you can review this slide on your own, but just showing you about more detail as far as elements in the periodic table. In each of the columns of the periodic table share the same number of electrons in their valence or outermost shells. So bonding. I'd like to think that we're doing a little bit of bonding this evening. We're talking about chemistry, but anyway, we're talking about chemical bonding, right? And so what we're looking at here is the different types of chemical bonds, right? So an electrical attraction that holds the atoms together. That's an important thing. So you'll see here that, oh, I went too far with that. There we go, right? So the types of bonds we have are ionic, covalent, and hydrogen bonding. Know that the hydrogen bonds are more of a weaker bond. Okay? And again, too, just discussing and referring back to the fact regarding that valence shell, right, that outermost shell. The number of electrons in the valence shell determines the likelihood that an atom will form a chemical bond. Understand? If it's unstable, then it's going to want to form a bond in order to then fill that outermost shell. Okay, so ionic bonds. So what did we say was an ion again? We have cations, positively charged, anions, negatively charged. So do opposites attract when it comes to ions? Yes, right, they do, right? So you'll see here that we have sodium chloride, C, right? You'll see it's examples of, so we have here from A to B to C as far as the positively charged sodium and the negatively charged chloride ion form to come about to form sodium chloride, table salt, okay? So we have the cation is the positively charged sodium, the anion, the negatively charged chloride ion. Ionic bonding, okay? Now, covalent bonds. Covalent bonds, you're gonna have equal or unequal sharing of electrons, okay? So let's see here. Covalent bonds formed by sharing electrons. Electrons are shared by the atoms. Now, polar or non-polar? What does it say here? Polar covalent bonds, and you're going to see it again in another slide. Polar covalent bonds, unequal sharing of electrons. Non-polar covalent bonds, electrons are shared equally. That's important for you to get that should really be something that you're going to be memorizing. And so here we're just taking this slide. I didn't highlight it here, I should have, but you're seeing here, next slide. Next slide. Non-polar, equal sharing of the electrons. See that? Equal sharing of the electrons. So you'll see here that the atoms, sorry guys. That's from Star Trek when like Kirk would like fight like a bad guy or something that would yes, yeah. Come on. All right, sorry gang. My brother. So equally sharing of the atoms, right? This is a non-polar covalent. Polar then is what? Unequally sharing of electrons. So think of the case of water, H2O. There's an unequal sharing of electrons in water. Okay, so think of, correct, yep. And so in water, the electrons are attracted more to the oxygen than they are to the hydrogen. So there's this unequal sharing of electrons, polar covalent bond, water. Now, but understand this, that, so water itself, H2O, let me go back here for a moment. This is an important point. Water itself, H2O, polar covalent electrons are unequally shared, unequally sharing of the electrons. But can we have molecules of water coming together? That's hydrogen bonding. You understand? So H2O, the bonds that are taking there are different than the hydrogen bonding of water molecules all coming together. Don't get that confused. Students get that confused. I want you to see that. So hydrogen bonds, what did I say to you that? They were what? Weak bonds. Weak bonds, yet still quite remarkable and such. I'll see next class if I can show you a little experiment with surface tension. Remind me about that, please. Surface tension. Show you that. So commonly occur between water molecules, hydrogen bonding. So the negatively charged region of a water molecule attracted to the positively charged region of another water molecule. See, these are all individual water molecules. Hydrogen bonding brings them together, but just one water molecule, H2O, polar covalent bond, its electrons are unequally shared. Right. So solutions. Water molecules are attracted to other water molecules and polar molecules. And so polar molecules and charged molecules dissolve to form solutions. And understand that water is amazing. It's remarkable. We are comprised of water, right? A good percentage of our bodies are water. And water, what do you think? Blood. What's the plasma in blood? Water, right? That's a major component of plasma is water, all right? We filter blood, we produce urine, right, in the kidneys and such via the nephron. And what's going on with that? We're removing waste products, and a good portion of that's leaving the body, it's water. But we reabsorb, and our kidneys will reabsorb like an incredible, a very high percentage, like 99% of what you're actually producing as a filtrate. We reabsorb that water and whatever else is present within, and only we're allowing a small percentage of the water to leave because we're trying to conserve whatever we have. See, the body is really good at recycling, honestly, and conserving what it has present within. So we only get rid of what we can't use, waste products. Understood? All right, so let's see here. So there, good, okay. So chemical reactions. All right, so let's go here. All right, so this first slide here is just going over different definitions that you need to know regarding energy is concerned, okay? And we're going to see that energy, the ability to do work. That's a very basic and simple definition. Doesn't matter where you see it in what textbook and such, basic definition, okay? Potential, kinetic, and chemical energy, they're all forms of energy, okay? So potential, we get potential. So it's some type of stored energy that has the potential to be used, right, for some type of reaction. Kinetic energy, energy of motion. Chemical, all right, potential energy stored in bonds. And we're going to see this in particular with ATP, adenosine triphosphate. We have three phosphates, and their bonds are connecting. And once we break those bonds between the phosphates, what's released? Energy is released as a result. Now, know this, that, and we'll talk about this as far as diffusion. So diffusion is the movement of from high concentration to low concentration. When we do this from high concentration to low, no energy is required. There's movement down that concentration gradient from high to low. Jazz hands, right? From high concentration to low. But if I have to go from low concentration to high, there needs to be some energy that needs to be used in order to do that, okay? So just have an idea. So that's an active process and such. But just remember that energy is required for many of the reactions in the body. Now here also you need to see exergonic, endergonic, okay? Exergonic reactions, they're releasing energy. Energy is exiting, it's releasing. Endergonic, it's energy is absorbing and it's entering into the reaction and such, okay? So I know it's very basic, and I am a terrible chemist, and I will admit that from the first day on. I'm a very good biologist, and I really know my stuff, but the chemistry aspect of it, it's always been a challenge for me. But just understand that, try to do the best I can with this chapter here for you all, okay? Just giving you the basics. All right, so here again, this is a, sorry gang, let's see, here we go again, here we go. So just giving you a reiteration regarding the different types of energy, potential, kinetic, chemical, talking about the bonds being stored, the energy stored in those chemical bonds. Next slide. So here we go. It's coming back to endergonic and exergonic, right? So energy can be stored in chemical bonds. We get that, I said that to you before. ATP is a common form of cellular energy, right? Where is ATP formed? Mitochondria of the cell. Very good. No, it's all good. It's all good. Mitochondria of the cell contain and will produce the ATP present that needs to be used, powering reactions and such. When we think of this, let me step back for a moment also, since I mentioned mitochondria. The nucleus of the cell and the mitochondria contain the DNA in the cell. Primarily, it's the nucleus of the cell will contain the DNA for the cell, right? But there's also going to be DNA present in the mitochondria, because if there's a need, the mitochondria can replicate and produce more mitochondria. So they have mitochondrial DNA, okay? Now, you'll see here, endergonic reactions require energy to store it in the bonds. Exergonic reactions will release that energy. So endergonic, entering into the reaction. Exergonic, it's being released, the energy, through those exergonic reactions. All right. Now, excuse me one moment. I apologize. Oh, is that okay? I know my brother was trying to reach me before. Let me just see if he. No, it's all good. It's okay. Okay. So when we think of reactions, reactions are made up of reactants, right? Those that will come together and react, And as they come together to react, they will then form, as a result of that reaction, a product. Does that make sense to you all, right? Simple enough. So the substances that enter into the reaction, reactants or substrates, the product is what's produced as a result of the reaction. Now, I've also underlined here and showed you that we have in bold print synthesis, decomposition, and exchange. So a synthesis reaction will have atoms, molecules, or ions combining to form a new molecule. So we'll have smaller molecules coming together to form what? A larger molecule. That'll be the product, okay? Decomposition, we're taking a larger molecule, right? And we're breaking it down. Decomposition, we're taking a larger molecule, breaking it down into its subsequent reactants and such. And so in the product, you're going to have, so the larger molecules broken down into smaller atoms, ions, and molecules for decomposition. Exchange is a combination of the two, exchange reactions, combination of synthesis and decomposition. And here we go into just giving you a little bit here, seeing that a synthesis reaction, we're taking reactants and producing a product, right? So endergonic, ATP is entering into it, and so then we're producing this product. Exergonic, we're taking a larger molecule, we're breaking it down. So what are we doing? We're breaking bonds. If I break a bond, what happens? ATP is released, right? Energy is released. Exergonic. And then the exchange reactions, it's a combination. So you're seeing on to the right side here, you're seeing examples of. Okay. Now, as far as what's going to affect how a chemical reaction will take place, well, the properties of the reactants, temperature, concentration, and pressure. And we're going to see that as we study the human body and the anatomy and physiology of it, in particular the physiology of what's going on, we're going to realize that pressure, that concentration is a big deal, and that concentration, having a difference in concentration, right? So look at this here. So if I were to put, or this, these represent sodium ion concentration, potassium ion concentration. Now, when we talk about the plasma membrane and what's taking place as far as polarity is concerned, we're going to see that as a result of concentration gradients, right? The difference between, if this represents the plasma membrane, right here, and outside of or inside, so intracellular or extracellular, and the concentrations of sodium ions and potassium ions they're going to be different extracellularly and intracellularly. And as a result, that's creating a polarity here. And the change in the polarity of the plasma membrane will allow for things to happen. I'm not going to get into details right now, but we're going to just know that concentration gradients means that there's going to be different concentrations of whatever we're talking about here. We're talking about potassium and sodium ions. There's going to be a difference. And so naturally, how will we move? Naturally, we're going to move from what? High concentration to low concentration, okay? Right. So I don't want to overwhelm you with a lot, but I'm just trying to give you enough to be dangerous, so to speak, but just to give you enough to give you a little bit of information regarding that. Now, enzymes primarily are they're proteins, okay? Proteins, and they will help to facilitate a reaction. And many reactions in the human body are facilitated by enzymes, which are proteins. And they're going to reduce the activation energy required for a reaction to occur. Okay, so enzymes really are very important in facilitating reactions. Right? And so that activation energy, it's the minimal energy required for a reaction to occur. So this is a good thing. So enzymes, and know this, that enzymes, when they're used in a reaction, they don't get used up. They can be used again, right? Which is really pretty remarkable, right? So they're able to be recycled and used over and over and over. So they're not used up during the reaction. So you'll see here, right? Let's go here. There we go. So enzymes lower activation energy. Enzymes are unchanged by reactions, and they'll be reused again over and over and such. So they facilitate reactions to occur. So you'll see here, inorganic compounds essential to human functioning. So let's take a look at the terms inorganic and organic. So you'll see here that inorganic compounds, they do not contain both carbon and hydrogen. Organic compounds contain both hydrogen and carbon. And when you think of organic compounds, what do I have down below there? Carbohydrates, lipids, proteins, nucleic acids. These are organic compounds. And if you have the pleasure of ever taking an organic chemistry course, it's very challenging, honestly. But it's very fascinating and interesting for sure. But it's that's a tough course for sure. But let's move on. So organic compounds, carbohydrates, lipids, proteins, nucleic acids. Here you're seeing as far as, so common chemical formulas in A&P, anatomy and physiology. So you're seeing here to the right, you're seeing here oxygen, O2, carbon dioxide, CO2. We know those both. We both know both of those, right? And so then we have potassium. We just looked at potassium. Sodium chloride, sodium ions, potassium chloride, potassium ions, chloride ions, calcium ions. And calcium ions, very important as far as when we talk about the muscular system and muscular contraction, as well as the nervous system. Calcium is very important. And you know what? What's quite interesting about calcium? And magnesium and phosphorus, absolutely. But think about, if I need a lot of calcium, shouldn't I have a place where I can go get it from? How about the skeletal system, right? It's a bank for calcium. So we need a lot of calcium in the body. And, you know, this is really important that we have the ability for breaking down of bone and replacing new bone, right? That's a process, remodeling and such. When we need more of it, calcium released, we're able to then release it into the blood as a result of breaking down old bone. So pretty cool stuff. You'll see glucose, you'll see water, H2O, you'll see hydrochloric acid, HCl, you'll see sodium chloride, NaCl. I said to you earlier there, as far as water, 50 to 70% of the adult body, right? So really that's quite a large percentage what goes on with water. And you'll see the functions, folks. Provides lubrication in joints. So think of synovial fluid. If the synovial joints are hollow, fluid-filled joints in the body, so think of your elbow, think of your knee, think of your hip socket, your shoulder. These are all synovial joints, phalanges in your joints in your fingers and your hands and your feet. So lubrication for joints, cushioning for cells and tissues and such. And understand this also, that present as far as for like think of the different cartilages and such. And so water is a component of the matrix, as well as other chemicals and such, to allow for that to have its ability to protect and to cushion. Aids in temperature regulation, right? We know this as far as sweat is concerned, right? Oops, go back. A solvent for ions and nutrients needed by the cells, involved in dehydration synthesis and hydrolysis reactions. And so with dehydration synthesis, you're going to end up dehydrating something. And what's one of the products from dehydration? Water, right? Because what are you doing? You're removing water from the reactants. There we go. Okay. So you're seeing here as far as solutions. And when we talk, we'll go more into detail with this regarding when we're looking at diffusion and such, and osmosis. We'll be talking about this, okay? And solutes in particular. So we'll go over that in more detail, and we'll be talking about that as far as for in the case of blood and the cells present, as well as other products present within the plasma of blood. We'll get into more detail with that. We'll move on. And concentrations of solutes, concentrations of the solvent, and this is going to be important as far as understanding how materials are moving in the body from one area to another and such. And we'll be going into more detail regarding this. And not only just liquids per se, right, but also gases and such, and other products we'll be looking at. You'll see here as far as different types of solution. So you have a colloid, you have a suspension. Here we go. Nope. Back one. Here we go. So the role of water in chemical reactions, I just mentioned this before. So dehydration synthesis, right? So forms new molecules while creating water at the same time. So what's left as a result of this reaction to occur? Water is also being released, okay? Hydrolysis reaction, we're going to be breaking bonds using water. Hydrolysis, a lysis is taking place. So breaking bonds, breaking covalent bonds, and we're using water to do this, and then we produce the products that are left over. Salts. So they're formed when ions bond through ionic bonding, right? Positive and negative forming, dissolve into separate ions when placed in water. So the dissolution that'll take place when placed in water. Now, this is an important aspect also that you'll have to get the foundational information understanding of. It's pH, okay, pH. And so know that, let me take you to the next slide in just one moment here. You'll see that acids release hydrogen ions, the H positive, right? It's a cation. Hydroxyl ions are what? They're negatively charged, anion. That's for bases, right? Bases will release this. Well, how can they release this unless they have an abundance of? They have more than, right? So an acid has a, look at this, an acid has a high concentration of hydrogen ions. A base has a high concentration of hydroxyl ions. Do you understand that? Does that make sense to you? How else is it going to be able to release them if it doesn't have a very high concentration of them? So that's when you look at this next slide here, what I want you to understand is that, so look here, so here's an acid, right? So battery acid, hydrochloric acid, pH of 0. That is the highest concentration of hydrogen ions present for an acid would be there, okay? From each, when we go from 7 to 6, 6 to 5, 5 to 4, they are, it's, so listen to this. 5, let's do 5. 5 is 10 times more acidic than 6, a pH of 6. Okay, let me repeat myself. A pH of 6 is 10 times more, well, no, sorry, a pH of 5, a pH of 5 is 10 times more acidic than a pH of 6. So it's by factors of 10 between each of the numbers in the scale. You understand me? So would a pH of 4 be then 100 times more acidic than a pH of 6? The answer to that is yes. Thank you. Appreciate that. So do you understand? So at each of those numbers, a factor of 10 between them. Understood? Now we go to the base side, basic alkaline, right? So then they are hydroxyl ion concentration. So at the very end of the scale at 14, that's the highest hydroxyl ion concentration. Does that make sense to you all? Okay. So pH scale from 0 to 14 indicates the acidity or alkalinity of a solution. Again, between each of the numbers, there's a 10 time, a factor of 10 between them. pH of 7 is neutral. Buffers are used to prevent rapid changes in the pH of a solution. And know that, like, really the human body here, we're looking at 7.3 blood. So a little bit more on the alkaline aspect, little further, not a full 8, right? By a factor of 10, but a smaller amount, 0.3. It's not good. So like cancer cells thrive in an acidic environment. It's not good. Well, I don't want to get, but here's the thing also, know this, so cancer cells thrive in an acidic environment. If, when we eat meats, that can lead to more of a lower pH. If we eat more vegetables, that can lead to more of a basic. So do you see how diet can affect also long-term what goes on with pH and such? So move on. Organic molecules, we said carbohydrates, lipids, proteins, and nucleic acids. Okay, so we're just going to go over some basics. And so carbon is a key. Carbon and hydrogen ions are key when it comes to organic compounds. And you're seeing here, this is just representation of the chains of the different diagrams of the organic molecules. Functional groups and such, I'm not going to deal with that right at this point in time because we're going to see that in your chemistry class. They'll talk in more detail regarding that. I'm not going to require information regarding this. So monomers, individual units that make up organic molecules. So foundational, this is what carbohydrates are produced via monosaccharides. They're in its basic form. Lipids, fatty acids, and glycerol. Amino acids, protein. So if we bring a bunch of amino acids together and bond them, what are we going to produce? A protein. Nucleotides for the nucleic acids. Carbohydrates, so we have mono is 1, di is 2, poly, many. Understood? So a monosaccharide, think a molecule like sugar, like glucose, individual sugar molecule. If we have disaccharide, so sugars like lactose are made up of what? 2 monosaccharides. A polysaccharide, there are many many monosaccharides bound together to form large, more complex carbohydrates. So the main source of chemical energy in the human body, carbohydrates. Examples of monosaccharides, glucose, fructose, galactose. Polysaccharides, starches, glycogen, cellulose. Understand this, that in the body, glucose is stored as glycogen. Liver, skeletal muscle. It's broken down, and what is it? What is it? If we break down glycogen, we release what? Glucose. Okay, so we take a polysaccharide, break it down, we're able to release then monosaccharides. Disaccharides, just two. Sorry. Yeah, here we go. Disaccharides, sucrose, lactose, maltose. 2 monosaccharides. Functions of carbohydrates used to produce ATP, right? That's a big deal. So primary source of cellular energy. Okay? Components of plant cell walls. Understand that the cell membrane, primarily it's phospholipid bilayer, okay? But carbohydrates help in that also and stabilize it. Let's see here. Lipids, fats. So nonpolar hydrophobic molecules. That means that it's not water loving, right? So it's not, so if it's hydrophilic, water loving, water is not an issue. But hydrophobic, it repels water. Okay. Major source, energy source for the cells, provides insulation. Fats, lipids. There we go. Saturated, unsaturated. Okay. So here we go. Solid or semi-solid at room temperature, saturated fats. Unsaturated fats, liquid at room temperature. Okay, so that just gives you an idea regarding that. Phospholipid, very important. Phospholipids compose that cellular membrane. So when we look at a phospholipid, an example of how you're seeing that there would be bilayer, 2 phospholipids, right? And you're seeing that the hydrophobic tails go inward. Hydrophilic heads exposed to extracellular environment, intracellular environment. Okay. Cholesterol, aiding in the stability of the cell membrane. Prostaglandins involved in inflammation and such. These are fats, lipids. Proteins. Proteins, we're taking amino acids, connecting them via peptide bonds, and we're producing proteins. And there are, as far as structurally, there are primary, secondary, tertiary, and quaternary structures for proteins. From the more simple to the more complex. Amino acids are the monomers of the proteins. We said that before. 20 amino acids in total. These are important. We have essential amino acids. We'll talk about this in more detail when we talk about the digestive system. I said to you again, linked by peptide bonds. That's what it says here. Again, showing you just an example of the structure of proteins from the primary to quaternary. Now, interesting here as far as seeing the term denaturation or denaturation, right? If I denature a protein, it's going to change the shape and will no longer carry out the function. So what about when I take an egg and I put it under heat, right? Will I denature the protein? Will I change the proteins? Absolutely, right? How about if I use an acid? Can I denature protein? Yes, you can. Absolutely, yeah. So that's acids and heat can all be ways that we can denature protein. Recall I said this before, that enzymes are proteins, okay? Enzymes facilitate reactions. Enzymes are not what? They're not used up in a reaction. So they speed up a reaction, they reduce the amount of energy required, that activation energy, increase the speed of a reaction. They're proteins and they're not used up. Nucleic acids. You all know these. You know DNA, you know RNA, you recognize those. Okay. So deoxyribonucleic acid and the monomers that DNA is composed of are adenine, cytosine, guanine, thymine. These are the bases. Okay. In RNA, you have adenine, cytosine, and guanine, right? Just like you do in DNA, except instead of thymine, you have uracil. And know that as far as bonding is concerned, adenine to thymine in DNA, adenine to uracil in RNA, cytosine to guanine in DNA, and again also in RNA. So what is it here? They store the genetic code of the cells and participate in protein synthesis. And so we'll look at this a little bit more in detail as far as talking about transcription and translation when we look at the next chapter regarding cells. And here you're seeing, as far as just what I mentioned to you before, the base pairing that takes place as far as during this whole transcription transcription, and translation process in order to eventually form proteins. So DNA, double-stranded, RNA is single-stranded. And you're seeing here the bases that are present. And I mentioned to you already where the base pairing is taking place. ATP, the energy currency of the cell. Okay, this is very important as far as ATP is one of the major components in order to, when energy is needed, ATP is what's there. Energy is released when bonds are broken. Like we said, as far as the phosphate, you see here, ATP, we break the bond, and then we have what? We have ADP and an inorganic phosphate that is separated from. And we'll talk about this in more detail when we look at, in particular, when we look at skeletal muscle contraction. Okay. So that was giving you a good idea as far as just a rough review. And again, this is not a chemistry class, but just giving you a rough idea as far as just some basics and highlights. So really just highlighted the chapter of what we really want you to know as far as from this class. Again, you'll all be taking chemistry classes, and so you'll, if you haven't already or you are currently taking, and you'll get more details regarding chemistry. But we will, again, talk more about in physiology and biochemistry what's taking place in the human body. All right, folks, so let me stop recording.